This video is going to look at transcription, and transcription is where we take the DNA, which has the genes on it, and we're going to convert that into RNA. Now DNA and RNA, specifically the type of RNA that we're going to be making is the messenger RNA. They're both nucleic acids, and we know that. They both have the Na as part of their name, but they're also going to have similar bases like the adenine and guanine and cytosine and so on. So the information from the DNA is copied over pretty easily into messenger RNA since they do have a lot of similarities, being both nucleic acids. This is going to be taking place in the nucleus of your cell because the DNA is in the nucleus, so anything that's related to the DNA has to be in the nucleus. Transcription allows your DNA to stay in the nucleus because transcription is going to be converting that information into messenger RNA, and the messenger RNA then can leave the nucleus and go out into the cytoplasm. So the DNA can be left in the nucleus where it is more protected, and that way if you were to ever have any cell invasions with bacteria, viruses, so on, it's just more protected to be in the nucleus, and so that's where we would like it to stay. The mRNA is the complement to DNA. So remember when we did our replication, we had the DNA and you were asked to make the complement strand to the DNA. Well, we're going to be doing something similar, only now we're going to be making the complement from the DNA to the messenger RNA. And what we need to remember here is that if you look back in your notes, RNA doesn't use the letter T. So whenever you have an A in your DNA, we would complement that with a T, but messenger RNA you can't have a T, so we'll be replacing that with a U. So this is what DNA transcription looks like. So this is this very large shadow here in the back. That's what we call RNA polymerase. And so this is another enzyme, and it's specifically an enzyme that helps this process of making messenger RNA just go a little faster. You have your DNA strand here, this blue, and it's the double helix, and then you have the single strand of red, and that's your messenger RNA. So mRNA is single-stranded, and notice how the mRNA and the DNA are bonding together. So we are making from the DNA what the complement mRNA nitrogen bases would be. This is just another picture showing you the same type of thing. So the RNA polymerase, like I mentioned, is going to be helping speed this process up, but some more specific jobs that it has, it is going to bind to your DNA and it's going to separate it, because remember your DNA is a double helix and it has the two halves of the ladder. We need to separate the DNA, we need to cut it so that we can access these nitrogen bases that are in the middle. And then it's going to use the DNA nitrogen bases to assemble what the complement RNA strand would be. The RNA polymerase can only bind to the DNA at a specific site, and that's called the promoter. That way, not 100% of your DNA is a gene. When we had the DNA in an earlier picture, we said that this might be a gene, gene number 1, and then down here maybe we have gene number 2. So you have a lot of like filler DNA here in the middle that isn't coding for a gene. So we want to start coding only in this gene section. We don't want to make a protein for something that our bodies don't need. So we're going to only be coding this gene into the messenger RNA. So there are going to be specific spots on your DNA that have a code on them that your body will recognize, and that's where it's going to bond so that it can start coding this particular region. And the DNA sequence will also tell it where to stop. Okay, so the promoter is the starting place, but there's also a code to let it know when to stop making this mRNA. So here's an example, and it's a very short example because we'll do some other ones in future videos that will be a little longer. But for now, just so that you can get an example of one of every letter. So these are the 4 letters that you have in DNA: T, A, G, and C. Now we just mentioned that RNA, because that's what you're asking to go to here, is this mRNA. mRNA doesn't use the letter T. Now T would normally pair with an A, and so it still does. Okay, T can go to an A. mRNA does have adenine in it. Here's where our difference is going to be. A will normally pair with a T. I'm going to write it in there small because mRNA does not have the letter T. It doesn't have thymine. So whenever you feel like, oh, I need to have a T and it's mRNA, that's where you want to put the new letter, the uracil. G will still pair with C, and C will still pair with G. So this is our new base pairing rule. When we have DNA, T goes to A to U, G to C, and C to G. So if you didn't write that in, make sure you do that now. And we're also going to use some new vocabulary as we go through transcription and then translation. And one of those words is called a codon. A codon is 3 mRNA nucleotides. So if I rewrite this so it's a little easier to see, AUCG. Every 3 nucleotides is called a codon. And it has to be in a row, so you can't have the A and then the C and the G and skip one. It has to be consecutive, meaning in a row, nucleotides. And that's going to be a codon. And eventually, when we get into translation in a future video, every single one of these codons will give us 1 amino acid. So that's just one piece of our protein. So here's your mRNA strand. We know that it's mRNA because it's single-stranded. And we have all of these bases here. So here we have wrote out all of the bases. Notice that we have no T's, so we're going to have U's throughout, but no T's. That's another indicator that this is messenger RNA. And they've broken it down so that you can see every 3 letters is a codon. Every 3 letters is a codon. So in order to have 7 codons, each one is 3 letters, so 7 times 3 would give you 21 nucleotides just to have 7 codons, not 7 amino acids. Some proteins are made up of like 500, 600 amino acids, so you're looking at like 1,500 DNA letters just to get that amount of amino acids. So it takes a large piece of DNA in order to do this. Alright, let's practice a little bit more. And we're going to be practicing here, we're going to be converting it to mRNA. So what I want you to notice is that I have added these lines, and I'm just going to put these lines between for our codons to kind of break them up so that we can see where the codons are. So if we have a T, we should know that T now goes to an A. We know that A can't go to a T anymore. It's going to go to a U. C goes to G. C again goes to G. G goes to a C. A goes to a T, but we don't do T's anymore, so it goes to U. And if you're if the line of letters is kind of long, you can always break these up into 3s as well, and that'll kind of help you not to get lost in the middle, because if you skip a letter, it could make things more difficult as we get into making our proteins. So for our next group of 3, we have T, T, and G. So we know the T's are going to go to A's, and the G will be converted to a C. And our last group of 3, we have C, C, A, so that would be G, G. Here you would normally put a T, but we don't have T's anymore in messenger RNA, we have U's. So we have that here. So it might be a little easier if I erased it so that you could see the actual computer letters. But that should be what your answer is. And each one of those, remember, each group of 3 is a codon. So in this particular example, our 12 mRNA letters, the 12 DNA letters, that only gives us 4 codons. And each one of these codons would be an amino acid. and so I'll abbreviate amino acids AA. You're only going to get 4 amino acids out of this. If this was your whole entire gene, you would only have 4 amino acids that make up that protein.